Welcome to the SANS Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. Before we jump into today's deep dive, please take just a moment to like this video and subscribe to our channel. It really does help support the SANS Science Experience, and let's just keep bringing you these explorations into, well, groundbreaking research. All right, so today we're diving into a genuinely groundbreaking scientific paper. It was published in Cell, which is, a top-tier journal. This work comes from the team of Jinlong Wai Lu and Juan Carlos Ispisua Belmonte. They're at Altos Labs. And honestly, this could fundamentally change how we think about aging and disease, too. They've characterized this newly identified, really widespread phenomenon they're calling mesenchymal drift, MD for short. So our mission today is to unpack this concept for you. We want to explore its surprisingly pervasive impact, across the human body, and reveal the truly exciting part, how this team found it might actually be reversible. We're going to try and make a complex scientific idea accessible, engaging, and relevant for you, offering a fascinating new perspective on health, disease, and maybe even longevity. Okay, let's unpack this then. Mesenchymal drift. What exactly is it? The paper calls it a cell state drift. Right. So imagine your body's cells. They're each typically designed for a specific job in a specific tissue, right? A lung cell, a kidney cell, whatever. With mesenchymal drift, these cells start to, lose their original specialized identity. They drift away from it. They begin to turn up the volume, basically, on certain specific genes called mesenchymal genes. Now, mesenchymal cells, they're usually involved in things like structural support, repair, forming new tissue, often related to scarring, actually. Exactly. So when other cells undergo MD, they start acquiring or sort of intensifying these mesenchymal traits. This means changes in how they produce the extracellular matrix that's like the scaffolding and glue around cells, also changes in their cellular junctions, how they connect to each other. And it affects tissue stiffness, how stiff the tissue around them becomes, and even what signaling molecules or cytokines they're pumping out.
Yeah, the paper makes it really clear.
MD isn't some small, isolated thing. It's a pervasive shift. And they call it a conserved hallmark of human aging. It seems to be happening across your body as you get older.
And what's truly fascinating here, I think, is how this drift isn't just some side effect of getting older. It seems to fundamentally contribute to organ dysfunction across the entire body. If you connect this to the bigger picture of aging, the research shows MD really goes hand in hand with other known hallmarks.
Yeah.
Right, like things we already talk about in aging research.
Exactly. We see a downregulation, a turning down of energy metabolism pathways. So cells get less efficient at making energy, and also cell cycle pathways, which affects how they divide and repair themselves correctly. And on the flip side, there's a clear upregulation, a turning up, of inflammatory pathways. This is what we sometimes call inflammaging, right? That sort of chronic, low-grade inflammation that builds up with age.
Ah, right, inflammaging.
Yeah. So understanding mesenchymal drift, it really offers this crucial new mechanistic insight. It helps us see the underlying processes driving aging at a cellular level. It's almost like finding a kind of master switch that influences many of these interconnected aging pathways together. That is a profound implication. I mean, suggesting MD could be a fundamental aging mechanism.
Oh.
But what really makes this paper, like you said, a game changer is how they connect this drift not just to general aging, but directly to how bad so many age-related diseases get. You mentioned the scale of their data analyzing gene expression from over 40 human tissues and, 20 diseases?
Over 20 diseases, yeah. It's impressive.
So what did they actually find there, and why is that so significant?
Well, they found mesenchymal drift is present in almost all of them, which really suggests it could be a unifying factor underneath it all. And it's not just there. Increased MD directly correlates with disease severity.
Meaning the more drift, the worse the disease.
Exactly. And also with reduced survival and overall increased risk of mortality. To give you a clearer picture, let's look at a couple of examples from the study. They're quite striking. Take the lungs. For conditions like Idiopathic Pulmonary Fibrosis, or IPF, that's a terrible disease where lung tissue gets scarred and stiff.
Yeah, devastating.
Right. In IPF, they see MD genes significantly turned up. And specifically, the lung's crucial air sac lining cells are called alveolar types. 1 and 2 epithelial cells, those decrease. While at the same time, scar-forming cells called myofibroblasts, which are driven by MD, they drastically increase.
So the functional cells disappear and scar tissue takes over, driven by this drift.
Precisely. And that's not just the lungs. Look at the brain. In Alzheimer's disease, they see MD gene upregulation in these really important support cells around the neurons. Cells like astrocytes, oligodendrocytes, pericytes. These normally help maintain brain health.
But now they're contributing to the decline instead.
It appears so. They're
Yeah.
showing this mesenchymal signature. And this pattern, this MD pattern, it's also seen across other major organs, the liver, the kidneys, the heart, consistently correlating with how severe the disease is. For instance, in advanced MASH, that's a severe fatty liver disease, MD goes way up. It really paints this picture of a systemic, body-wide problem linked to aging and disease.
That's a powerful and frankly, yeah, quite an alarming picture of systemic impact. And to really grasp the gravity, you mentioned the IPF data. Patients with idiopathic pulmonary fibrosis. The study grouped them based on their MD gene levels, right?
That's right, lowest versus highest levels.
And those with the lowest MD expression had a median survival of over 1,000 days, 1,027 days. But those with the highest levels, they survived for only 294 days.
Just 294.
Let that sink in. That's more than a 40-fold difference in survival time, just based on the cellular signature.
Yeah.
That's not just a marker, that feels like a profound indicator of how devastating this drift really is. But what's the reason? Why does this drift happen? Why do our cells start losing their identity like this?
Well, if we try to connect this to the bigger picture again, the paper suggests MD might arise from repeated cycles of tissue injury and repair. Things our bodies go through all the time throughout life. Every cut, every infection, every bit of stress can trigger some kind of repair process.
Okay, so wear and tear, basically.
Sort of, yeah. And as we age, there's also accumulating damage to these long-lived proteins and the extracellular matrix, that scaffolding we talked about. That damage often leads to tissue fibrosis, scarring.
Right.
And crucially, they found that certain circulating factors, like members of the TGF-β family, these are known to increase during aging and are linked to mortality risk. These factors act as potent inducers of MD.
So things floating in our blood can actually trigger this drift.
It seems so,
Right.
and that could potentially allow MD to spread systemically, from one tissue to others, contributing to that widespread effect. It's almost like a signal telling cells, hey, forget your normal job, start acting like a repair crew, even when maybe that's not helpful anymore or goes too far.
Okay, so if this cellular drift is such a core problem, maybe a foundational mechanism for aging and disease, what does this all mean going forward? This is where it gets really exciting, I think. The most hopeful part of this deep dive, the potential for reversal. The paper explores this technique called partial reprogramming. It uses those powerful molecules, the Yamanaka factors, OCT4, SOX2, KLF4, and MYC-OSKM, the factors that can turn adult cells back into stem cells.
Exactly. And these OSKM factors, they've shown rejuvenating effects before, but this study gives us a much clearer idea of why. The key finding here is that partial reprogramming can markedly reduce MD in cells. And importantly, it happens before the cells fully de-differentiate into that pluripotent state.
Meaning they don't become potentially dangerous embryonic-like stem cells.
Right, that's a critical safety aspect.
Yes.
For example, they took aged human fibroblasts, those common connective tissue cells. Just 3 days of OSKM induction led to a really robust downregulation, a turning down of key mesenchymal genes, genes like SNAI1 and ZEB1, and even TGF-β genes were turned down. It effectively reversed the transcriptomic aging changes.
So it restored the youthful pattern of gene activity.
Pretty much, yeah. And what's even more compelling, they tested a mutant Yamanaka factor, OCT4YR. This one cannot induce full pluripotency. It can't make stem cells. But it still successfully suppressed MD and reversed aging signatures in the aged cells.
Wow, okay.
Which strongly suggests that knocking down this mesenchymal drift is a primary mechanism for the rejuvenation they're seeing. It's independent of creating actual stem cells. It seems less about turning the clock all the way back and more about restoring a healthy functional identity to the cell.
And this wasn't just in cells in a dish, right? They looked in animals, too.
Yes, the findings extended to living organisms.
Yeah.
In naturally aging mice, long-term partial reprogramming significantly decreased MD gene expression in organs like the kidney and liver. And those are organs known to get fibrotic, scarred as they age. Even shorter-term treatments showed positive trends in tissues like the spleen and liver. So this suggests partial reprogramming can actually transform that stressed pro-aging secretory profile.
Basically, stop the aging cells from sending out bad signals.
Exactly. Stop them from sending signals that promote inflammation and damage and shift it towards a more youthful state. And this seems to lead to overall improved health and tissue function in the mice. The implications are, well, they're enormous.
Yeah, absolutely. This deep dive really highlights mesenchymal drift as potentially a fundamental mechanism driving both just normal aging and also the progression of so many age-related diseases. And the fact that it seems reversible through partial reprogramming, that offers this really compelling, exciting framework for maybe developing future interventions. You can imagine therapies that could directly target this cellular drift, kind of re-anchor cells to their healthy, youthful identity.
It's definitely exciting. But this raises an important point, too. While the findings are incredibly promising, the study authors themselves point out some limitations, things future research really needs to tackle.
Okay, like what?
Well, for one, they say more in-depth epigenomic analyses are needed to fully understand the underlying chromatin mechanisms that drive this MD reversal.
Epigenomics, that's about the tags on DNA that control genes, not the DNA sequence itself, right? And chromatin is how DNA is packaged.
Exactly. So we need to understand those deeper layers of gene regulation to see how reprogramming fixes the drift. Also, while the results in human cells and mice are great, the study used a relatively limited number of human donors.
Oh, okay. So we need bigger studies.
Definitely. Future research needs larger, more diverse groups of people to make sure these findings apply broadly, and eventually to figure out how to personalize therapies based on this.
Right, makes sense. Still early days in some ways, but incredibly promising direction. Okay, so here's something for you to mull over. If this drift of our cells away from their young, specific identities is a core driver of aging and disease, what new possibilities really open up when we think about re-anchoring them, finding ways to pull them back to that original state? The future of tackling aging and disease might truly lie in restoring cellular identity. Thank you so much for joining us on this deep dive into the latest in science, and please don't forget to like this video and subscribe. It really helps support the SANS Science Experience. Join us next time on the SANS Science Experience.